Hello everyone. Hope you all are doing fine. This lecture is about host pathogen interactions. This lecture is useful for the students of microbiology, medical lab sciences, MBBS, BDS, public health, and nursing, and for everyone who wants to know how infection happens. Before we start, I, Dr. Tripti Malik Ahuja, welcomes you to this channel. If you are new to this channel, then do subscribe it and don't forget to press the bell icon so that you can get notifications of the new videos. In this figure, you can see these are two pathogens who are fighting against the host so as to defeat this host. We will see this process and we will learn the complete process of the infection in detail in this lecture. So watch this video till end. Let us revise terminology related to the infection. Pathogen is defined as an agent capable of causing infection. These are usually microscopic. While the host is larger living organism, it can be infected by the pathogen or fed upon by the parasite. The infection is defined as the entry, invasion, and multiplication of the microorganisms or can also be called pathogens such as bacteria, viruses, fungi, and parasites that are not normally present within the body. I have described these terminology in the previous videos. You can get the link from the description box below and from the I button here. The pathogen and the host. They interact with each other and may result to the infection. The outcome of this interaction process depends upon the strength of both the host and the pathogen. Infection is a multi-step process. These steps are followed one after the other. And sometimes these steps they can be concurrent, that means they can occur side by side also. The first step is the entry, or we can also call it transmission. The next step is a crucial step of attachment or adherence. Third step is of the colonization. The mitocolony of pathogen is formed usually at the entry site. Next, fourth step is the step of invasion, in which there can be penetration of the pathogen in the deeper tissues. The pathogen survives in the host body provided it is not eliminated or acted upon by the host defense. Finally, there is injury or damage to the body of the host. So all these steps they occur in sequence. Hence, this situation it can be compared to like the battle like situation. The infection can be studied in the form of a battle. We can see that there are two opponents in this battle. A battle is going on. This one is the intruder who is trying to enter in the neighboring country so that it can cause the nuisance there. The pathogen is considered as intruder here. The pathogen is carrying weapons with it. These weapons are called the virulence factors of the pathogen. The second opponent is the host. Host is like army of the neighboring country. The host is having a defense mechanism in the form of the immunity. The outcome of this battle, this interaction between the host and the pathogen, it depends on the strength of the virulence factors of the pathogen And it also depends upon the level, the strength of the defense of the host. So, who will win this battle? That depends whosoever is strong. If pathogen is strong, then the pathogen is going to win. The pathogen will cause the infection. If the host is strong, then the host is going to win and there will be protection, there will no infection. The first step is the step of the entry in the body of the host. For the entry in the body, the pathogen has to enter through the portals of entry. These portals of entry are the mucosal surfaces or the skin. The different routes through which the pathogen that can give entry are the ingestion, inhalation, trauma, needle stick and injuries, catheter, the orthopod bite, or the sexual transmission. So in this figure, the different portals of entry they are shown. The portals of entry are shown with the arrow which are pointing inwards. The mouth, respiratory tract, urogenital tract, conjunctiva, the entry via the skin, through the scratch, injury, orthopod, or through any sharp instrument can be there. The next crucial step, the second step, is the step of the attachment or the adherence. It is the prerequisite step of the infection process. If the pathogen doesn't attaches or adheres, then the pathogen will be washed away by the flushing action of the body fluids. So a pathogen has to attach, it has to adhere. For this, the pathogen, they have got the virulence factors. These virulence factors, they are called the adhesins, or the pathogen can also attach through the hydrophobicity. The adhesins, they are present on the surface of the pathogen. Here we are considering the pathogen to be bacteria. And because of this adhesin, the pathogen, they bind to the receptors. The receptors are present on the surface of the host cell. There has to be interaction between the virulence vector of the pathogen and the receptors present on the surface of the host cell. The adhesins, which are the virulence factors responsible for the attachment, they can be classified as fimbrial adhesins or the afimbrial adhesins. Fimbrial adhesins, they are present on the fimbriae or the pili. Afimbrial adhesins are the adhesins other than the fimbriae. In this figure, we can see there are different bacterial structures which are involved in adhesion to the host cell. This figure describes the different adhesins which can be present in the bacterial cell. A bacteria can have all of these or one or more than one of these adhesins present on its surface. The most common one are the fimbriae. The afimbrial adhesins, they can be fibrils, flagellum, capsule, as layer, and cellular. The type of fimbriae or the fimbrial adhesins. The fimbriae, they have got the adhesins located either at the tip of the fimbriae or along the length of the fimbrial shaft. The fimbrial adhesins, they are present commonly in the gram-negative genera, Bordetella, Salmonella, Neisseria, Yersinia, Pseudomonas, or Phytomonas. There can be five major types of the fimbriae, the type 1, type 2, 3, 4, and 5. These fimbriae, they differ in the respect of the rigidity or flexibility or on the basis of the process to agglutinate the RBC cells. In addition to this type 1 to type 4, 5 fimbriae, there can be the S. pili, X. pili, the PAP. pili, pyelonephritis associated pili, or Dr. fimbriae present in the bacteria which can function as the adhesins. For the afimbrial adhesins, the bacteria can have flagella. Flagella is present in the bacteria Pseudomonas aeruginosa, Helicobacter pylori, Campylobacter jejuni. The capsule can also be called glycocalyx or slime in other cases, is present in Streptococcus, Klebsiella, and Bacteroides. As layer, they are the array of proteins present in the cell wall, present in bacteria such as Aeromonas, Salmonellida, Lactobacillus acidophilus. Some specialized structures they are also present for the attachment. For example, mycoplasma. It has the surface structures P1, P13. Treponema pallidum, they have the surface adhesins P1, P2, and P3, because of which it binds to the fibronectin of the host cell. In addition to these, there are some molecules present in the cell wall, such as the leptins, carbohydrates, lipopolysaccharide, teichoic acid, outer membrane proteins, and enzymes, which can also have the function of attachment. So they are also classified under the afimbrial adhesins. This was all about the first step of the attachment, which is mediated by the virulence factors called the adhesins. The consequent steps of the invasion and the damage to the host body will be continued in the second part of this lecture. So stay tuned. All the best for your future. Bye and take care.